Welcome to my YouTube channel Science World. Today we are going to talk about the photosynthesis, which is the fundamental process that powers life on Earth. So let's see what is the process of photosynthesis. Photosynthesis is mainly happening in green plants, algae and some bacteria. In this process, the light energy is converted into chemical energy. This process is mainly happening in the chloroplasts, which are having chlorophylls. So you can see the structure of chloroplast in here. In this structure of chloroplast, you can see an outer membrane, an inner membrane, and also an intermembrane space. The intermembrane space is the space in between outer membrane and inner membrane. Also, inside of the inner membrane, you can see a stroma. In the stroma, you can see grana and thylakoids. Grana is a structure prepared by few number of thylakoids. That's why grana is known as stack of thylakoids. In here, in the process of photosynthesis, there are two main stages. In here, you can see those two main stages, which is the light dependent reaction and the light independent reaction, which is known as Calvin cycle. Both light dependent reaction and light independent reactions are happening in the chloroplast. So we will see what is the light dependent reactions. As the name implies, light dependent reactions will be dependent on the light. So it requires sunlight, and also this will be happening in thylakoid membranes. Also, you can see the thylakoid membrane in here, and inside of thylakoid membrane you can see the thylakoid lumen, and outside of the thylakoid membrane you can see the stroma. In the thylakoid membrane you can see two special structures known as photosystem 2 and photosystem 1. Other than those two special systems, some special molecules known as plastoquinone, cytochrome B6, plastocyanin and ferredoxin, then ferredoxin NADP reductase and ATP synthase can be observed. In the photosystem 2, the electrons absorb light in the presence of light and transfer to higher energy stages. Then those electrons with high energy are unstable in those high energy states, and they will be come back to the lower energy stages by transferring back that energy, by emitting that energy, and those emitted energy will be transferred to the plastoquinone. Then again the electrons in plastoquinone molecule absorb those energy and go to higher energy stages. Again the same process will be happening, and those electrons in higher energy stages come back to the lower energy stages and emit energy. So according to this process, the energy will be transferred from photosystem 2 to plastoquinone, then cytochrome B6 complex, and then plastocyanin, and to the photosystem 1 and ferredoxin, then to the ferredoxin NADP reductase enzyme. This electron transferring process or the chain is known as the electron transferring chain. So because of electron transferring from photosystem 2 to ferroxine NADP reductase, which is attached to the outside of the thylakoid membrane. There is a demand for the electrons in photosystem 2. In the presence of light, the water molecules will be split into oxygen H plus and electrons. By the coming electrons from splitting of water molecules will be helped to complete the demand of electrons in photosystem 2. And also the H plus ions will be transferred to the stroma from thylakoid membrane through the ATP synthase protein molecules, which are having channels. So at the outside of thylakoid membranes, NADP, which is known as nicotinamide adenine dinucleotide phosphate, accepts the upcoming electrons and hydrogen ions from the stroma and become NADPH, which stores energy in the form of high energy electrons by reducing. So in here NADPH is formed, and in the stroma of chloroplast, adenine diphosphate molecules capture H plus ions coming from thylakoid lumen and another phosphate and become adenosine triphosphate, which is known as ATP. So in the light dependent reactions, ATP and NADPH are produced, and also due to splitting of water in the presence of light, oxygen is also produced as a byproduct. So up to now we have discussed what is happening in light dependent reactions in chloroplast. Now we will see what is the light independent reaction, which is known as Calvin cycle. As the name implies, for these light independent reactions do not require light, and it occurs in stroma of chloroplast. In the process of Calvin cycle or the light independent reaction, there are three stages as carbon fixation, then reduction of glyceraldehyde triphosphates, and the regeneration of RUBP, which is known as ribulose 1,5-bisphosphate. So the Calvin cycle contains three stages as carbon fixation, reduction of 3-phosphoglycerate, and generation of ribulose 1,5-bisphosphate. So in the first process of carbon fixation in Calvin cycle, 3 molecules of ribulose 1,5-bisphosphate captures 3 molecules of carbon dioxide in the presence of Rubisco enzyme and prepared unstable compound containing 6 carbons. Due to the instability of this compound containing 6 carbons, it will be immediately break down into 2 molecules of 3-phosphoglycerate and prepared 6 molecules of 3-phosphoglycerate. Then in the second stage of Calvin cycle, which is known as reduction of 3-phosphoglycerate, those 6 molecules of 3-phosphoglycerate will be reduced into 6 molecules of glyceraldehyde triphosphate. For this process, energy will be supplied by 6 ATP molecules and H plus will be given by 6 NADPH molecules. Then, in the final stage of Calvin cycle, which is known as regeneration of RUBP, among those 6 molecules of glyceraldehyde triphosphate, 5 molecules will become ribulose 1,5-bisphosphate. In the process of this regeneration of ribulose bisphosphate, totally 3 molecules of ribulose bisphosphate are produced. For this regeneration process, 3 ATP are needed to provide energy. And since from the 6 molecules of 3-phosphoglycerate, 5 molecules are helped to regeneration of ribulose bisphosphate, another 1 molecule of glyceraldehyde triphosphate will be helped to generate glucose, and in here half molecule of glucose is prepared. This process of light independent reaction or the Calvin cycle will be occurs in stroma of chloroplast. And for this process, as you can see, carbon molecules, ATPs and NADPH are used, and glucose is produced in this Calvin cycle. So let's see the formula of photosynthesis. In the photosynthesis process, 6 carbon dioxide molecules reacts with 6 water molecules in the presence of light in chlorophyll and produces glucose as main product. And also the 6 molecules of oxygen are produced as the byproduct in this reaction. This photosynthesis process only happens in the presence of light and chlorophyll. And also carbon dioxide molecules are absorbed from the air and water molecules are absorbed by roots from soil. Light is the energy source and chlorophylls are the green pigments which capturing light. So this is the formula of photosynthesis. Now we are going to see what are the factors affecting for the process of photosynthesis. The first one is light intensity. If we have more light, the rate of photosynthesis will be faster up to a certain point. The second one is concentration of carbon dioxide. If the carbon dioxide concentration is higher, the rate of photosynthesis will be faster. Third one is the temperature. Under too much high temperatures and too much low temperatures, the rate of photosynthesis will be reduced. This is happened because in too much high temperatures and too much low temperatures, the enzymes will be destroyed. Enzyme activity will be destroyed. So the last factor is water availability. Water is a main reactant for this photosynthesis process. The photosynthesis process can be experimentally observed by submerging an aquatic plant like Elodea in the presence of light and by observing the bubble production or the oxygen production. In here you can see an aquatic plant is submerged in water container, and the number of bubbles or the volume of gas prepared as a side product of photosynthesis will be measured. Then the rate of photosynthesis can be calculated by dividing the number of bubbles per unit time or the volume of gas produced per unit time. And also the gas produced by the photosynthesis process can be identified by relighting of a glowing split, as you can see in this video. So this is the end of our video. Hope you have learned and joined. Be with us in future videos with Science World. Thank you.